as Assistant Professor in the Department of Botany, Government College, Women University, Faisalabad, Pakistan. The topic of today's presentation is copper, cadmium and nickel pollution inhibit growth and promote ascorbate catabolism in cell cultures of Arabidopsis, thaliana and Ziamase. And I have performed this experiment under the supervision of Professor Dr. Stephen C. Fry, who is a serving professor in the Edinburgh Sullivan Group Institute of Molecular Plant Sciences, the University of Edinburgh, United Kingdom. And a part of research I am going to share with all of you. Regarding abstract, we are actually established model system for exploring the role of symplastic and apoplastic ascorbate in heavy metal fluted dicot and monocot cells. And in current experiment, we have used Arabidopsis as a monocot model plant and maize as a dicot model plant. And we have treated both plant species with copper, cadmium and nickel, and we have checked the growth and ascorbate metabolism in both plant species. And the cell suspension cultures of both plant species respond to the heavy metal stress by maintaining its symplastic ascorbate concentration, which may beneficially scavenge symplastic reactive oxygen species. Alternatively, apoplastic ascorbate is also catabolized in metal fluted cultures via several oxidative and non-oxidative reactions, the former potentially scavenging stress-related apoplastic reactive oxygen species. Methodology. First of all, we have actually prepared former salts of cadmium, nickel and copper. We have taken the chemical cadmium oxide, nickel sulfate and the copper chloride, and first of all we have dissolved these chemicals in the formic acid and then re-dried in vacuum dryer, and then we have dissolved the pellet in excess amount of deionized water to remove excess amount of formic acid, and lastly we have prepared the 20 millimolar stock solutions of all copper, cadmium and nickel solutions, and finally we have also confirmed the concentrations of all these three metals by high voltage paper electrophoresis. And for this experiment we have used cell suspension cultures of maize and Arabidopsis, and we have actually treated the cell suspension cultures of maize and Arabidopsis as a mini culture tubes having different metal concentrations for copper, cadmium and nickel. In this present picture you can see the different concentrations of the different metals. Actually we have tested the same concentrations for all different forms of the metals, but here just we have showing the comparison of different concentrations of different metals which are going to have a toxic effect to the Arabidopsis cultures. Here you can see a complete methodology is actually given in this section, and we have measured the growth parameters of both plant species, maize as well as Arabidopsis. The cell susceptibility to compaction by centrifugation was calculated by using formula laid down by the Fry and Street in 1980. And for fresh weight measurement, we have taken the same mini culture, treated or not treated with the different metals. They have filtered through the mirror cloth and gently squeezed with uniform pressure and then weighed to determine the change in their growth pattern with and without different forms of metals. And then we have also measured endogenous and the exogenous ascorbate concentrations in both plant species. Firstly, with the endogenous ascorbate concentration, that is going to have endogenous production of the ascorbate with and without metal, and then we have also used 1 millimolar concentration of ascorbate to be supplied to all the samples, either treated with metals or not treated with metals, and the ascorbate concentration was actually determined by using DC pip method, and that DC pip method is a very authentic method to determine either endogenous ascorbate concentration or exogenous ascorbate concentration. And then we have also used radioactive ascorbate, and that radioactive ascorbate concentration is determined qualitatively as well as quantitatively, and quantitative determination has been carried out by the scintillation counting, and while the qualitative determination of all the metabolites of ascorbate catabolism is determined by high voltage paper electrophoresis. And some very astonishing results we have received from this current experiment. Firstly, we have actually selected the most suitable concentration of copper, cadmium, and nickel for our both plant species, and we have actually taken a concentration from 0 to 2048 micromolar concentration, and that micromolar concentration from 0 to 2048 was determined in terms of their growth pattern for increase in the packed cell volume, and we have also calculated the susceptibility to compaction by using the formula as I have discussed in methodology section. And you can see the concentration of copper is quite drastic, that near about 128 micromolar concentration was quite toxic, and it is going to have a 50% growth inhibition at this concentration, while nickel is not that toxic at the same concentration. In fact, it is going to have some healthy cell suspension culture, and it is not going to affect that much on the growth pattern in terms of packed cell volume, and it is going to halve its concentration above 1000 micromolar concentration. And yes, cadmium is also going to have a similar effect on both plant species, as you can see, having 50% growth inhibition, about 500 plus micromolar concentration, while in maize cell suspension culture is going to tolerate cadmium and nickel a bit longer, about 1000 micromolar concentration, and we can see the toxic effect of cadmium and nickel uh, at more than 1000 micromolar concentration. And susceptibility to compaction, that is going to be increased with the increasing concentration of all the three metals. And not only that, we have also seen some different changes in change in the color of both plant species, Arabidopsis and maize, and we know Arabidopsis are green in color naturally, and maize are creamy white color in naturally as an healthy cultures. But with the toxification of copper, cadmium, and nickel, they are going to change their color, and you can see the positive three positive signs here are going to show the healthy cultures, and going down up to the 512 in both Arabidopsis and maize, they are going to cause some toxic effect by changing from green to yellow. And similarly, the maize color is going to change from creamy white to the brown. So here we have been not testing these concentration against as cadmium and nickel in both Arabidopsis and nickel for 8 micromolar, 16 micromolar concentration, and similarly 1024 and 2048 micromolar concentration was not being tested for this particular experiment, as we have seen previously in growth pattern that copper is going to have a toxic effect even at a very small concentration. So the further concentration were not being tested, and the earlier concentration for the cadmium nickel are not going to cause any toxic effect on both plant species, so these two concentrations were not been screened for the further experiment. And we have also seen some time course experiment for the growth of the metal fluted mini cultures, as you can see for the fresh weight and the packed cell volume of both Arabidopsis and maize, with an interval of 20 hours each, up to 0 to 160 hour for the both plant species and for both parameters, fresh weight and packed cell volume. And you can see the copper is going to decline fresh weight immediately, and we are going to have a very drastic growth reduction with the higher concentration of copper, while the concentration of nickel up to 512 micromolar is not going to affect that much. And similarly, in maize culture, you can see the 512 micromolar concentration of nickel is not going to have a negative effect on the growth pattern of maize culture, but certainly the 2048 micromolar concentration is going to affect maize culture for nickel and cadmium, and copper is going to be very drastic at a concentration of 512, and more or less similar findings were being exhibited for the packed cell volume. And regarding the endogenous ascorbate in metal polluted mini cultures, we can see we have again a time course study where we are going to have an interval of 20 hours where and up to 60 hours in both plant species Arabidopsis and maize, and all cultures are being treated or not treated. The not treated serve as control and treated ones as copper 2 concentrations, cadmium 2 concentrations, and the nickel 2 concentration for both plant species. And you can see particularly for the base, both nickel concentrations are going to have a higher ascorbate concentrations with the passage of time up to 160 hours, and while ascorbate is going to be catabolized or it is going to be diminished from the cell suspension cultures with the passage of time that were treated with copper 512, and it is going to sustain up to 80 hours in those cultures treated with the copper 128 in both plant species, as you can see. But certainly we can see a higher ascorbate concentration in maize cultures comparison to those in the Arabidopsis cultures. And certainly we need to check that the ascorbate that is going to be catabolized, where it is going to be. So firstly, here you can see the red arrows that are going to show this ascorbate is going to be oxidized into its further metabolites like semi-dehydroascorbate, then into dehydroascorbate, and further oxidation process it is going to be metabolized into the oxalyl threonate, cyclic oxalyl threonate, oxalate, L-threonate substances metabolites, and dash arrow lines are actually been showing non-oxidative conversion of these L-ascorbate into their further metabolites. And here is also a time course study, not in a long span. We are actually going to perform this experiment from 0 to 120 minutes with an interval of 20 minutes each. And here we have not only use like we are using the exogenous ascorbate concentration are going to be determined, and certainly we have taken the sample not only after 20 minutes, we have also taken the sample about 3 minutes, 5 minutes, 10 minutes, 15 minutes, and up to 20 minutes. So the sample has been taken periodically at different time intervals, and ascorbate is going to be catabolized. It's a exogenous ascorbate, as you can see over here. So this is going to be catabolized very quickly, very sharply in both Arabidopsis and maize culture which were treated with copper. And on the contrary, the nickel is going to support ascorbate um, catabolism to some extent up to 60 minutes, and then it is going to be catabolized very sharply. And again, we have also determined extracellular ascorbate that was radioactively labeled, and its metabolites which were not actually taken up by the cell, nor it has been going to be converted as carbon dioxide which has been released. So it is going to be determined by scintillation counting, and the similar pattern is going to be shown by the scintillation counting. And here we have and qualitatively determine the ascorbate catabolism by high voltage vapor electrophoresis. In conclusion, we have determined the presence of the metal line tested here at growth inhibitory concentrations. The cell, while ever they remained alive, appear to prioritize the maintenance, that is biosynthesis and or regeneration of their ascorbate, purposed to serve as an antioxidant that scavenge heavy metal generated reactive oxygen species. Apoplastic ascorbate was metabolically labile, that is half-life approximately 1 hour. In healthy cell cultures, it was more rapidly oxidized in the presence of many of the metal stress conditions tested, indicating that it served to scavenge reactive oxygen species. The nature of the extracellular ascorbate oxidation product detectors suggested that the major apoplastic reactive oxygen species generated in the presence of copper, cadmium, or nickel was superoxide rather than H2O2, that is hydrogen peroxide. For this work was actually gives a wonderful understanding about heavy metal toxicity and the role of ascorbate, and by highlighting these differences between the maize and Arabidopsis, it emphasizes that more research is required on different plant taxa so that we can generalize our findings more appropriately. uh, any question, according to my information, I would love to satisfy your query. And again, thank you, the International Organization Science Father, to nominate me as International Research Awardee on Organic Chemistry. Thank you so very much.